Welcome to the deep dive. Today we're tackling a topic that is just fundamental to life itself. The body's astonishingly precise regulation of hydrogen ion balance.
Absolutely. And if you really want to master how your body works, I mean, at a truly foundational level, this is key.
Right. We're talking about understanding not just what happens, but why it has to happen
Yeah.
with such incredible precision.
Exactly. And we're diving deep into the sources provided today. It's a really comprehensive look at the mechanisms involved so we can all get a proper handle on this vital system.
So why is it so crucial?
Well, because virtually every enzyme system, every single cell function is profoundly sensitive to the concentration of free hydrogen ions, the H plus ion.
Tiny shifts can just throw everything off balance.
Completely. Think about it this way. Compare it to an ion like sodium. Sodium's everywhere in your extracellular fluid, right?
Right.
Around 142 milliequivalents per liter.
Okay.
But the normal hydrogen ion concentration, it's minuscule. We're talking 0.000004 milliequivalents per liter, or 40 nanoequivalents per liter.
Nanoequivalents. So H plus is literally millions of times less concentrated than sodium.
Millions of times. And yet the normal range of variation for H plus is incredibly tight. Maybe only about 3 to 5 nanoequivalents per liter difference is tolerated.
Wow. Sodium can fluctuate way more than that without immediate chaos.
Exactly. So this extreme precision for something so dilute, it just shouts its importance, doesn't it?
It really does. It tells you this specific ion needs to be controlled with a level of vigilance we just don't see for most other things.
Right. To maintain life within those narrow margins, it's absolutely essential. Understanding this control is, well, it's fundamental to understanding physiology.
So our mission in this deep dive, then, is to unpack this high-stakes balancing act. We're going to go through the body's defense systems layer by layer, using the source material as our guide.
Yeah, aiming for that expert level of understanding, because this topic, it really underpins so much of clinical medicine, especially in critical care. Getting these fundamentals right is just essential.
Okay, let's start by anchoring ourselves with some core definitions. We keep saying hydrogen ion, H plus carb. What exactly is that?
It's simple, really. A hydrogen ion is just a single free proton. That's it.
And acid.
An acid is any molecule that can release one of those protons, that H plus ORF, when it's in solution. So like hydrochloric acid, HCl, it readily splits into H plus and Cl.
Right.
Or a weaker example is carbonic acid, H2CO3. It can release H plus and bicarbonate, HCO3.
So acids are H plus donors. What about bases?
Bases are the opposite. They're ions or molecules that can accept an H plus group. Bicarbonate, HCO3, is a really key base. It grabs an H plus to form H2CO3.
Ah.
Phosphate ions, like HPO4 double minus, they can accept an H plus to form H2PO4 minus. And importantly, proteins act as bases, too.
Proteins? How so?
Many proteins in your body have negatively charged groups on their amino acids, and these can bind to H plus ACTs. Hemoglobin in red blood cells is a huge one, and lots of intracellular proteins.
Got it. And the term alkali, is that different from base?
They're often used interchangeably in this context. Technically, an alkali refers to a specific type of strong base, usually formed from an alkali metal like sodium, combined with a very basic ion like hydroxide, OH. These react really fast with H+, but for our purposes, base and alkali often mean the same thing, something that accepts H plus K, and related terms you'll hear are acidemia, meaning low blood pH or high H plus Psi, and alkalemia, meaning high blood pH or low H plus Psi.
And acidosis and alkalosis.
Those refer to the underlying processes that are causing the acidemia or alkalemia. So acidosis is a process leading towards low pH. Alkalosis is a process leading towards high pH.
Right. And you mentioned strong versus weak acids and bases. Let's clarify that distinction.
Sure. Strong acids, like HCl, dissociate almost completely and very quickly, releasing a lot of H plus Psi. Strong bases, like hydroxide, OH, grab H plus really vigorously.
And weak ones.
Weak acids, like carbonic acid, H2CO3, they hold onto their H plus more tightly, releasing it less readily. Weak bases, like bicarbonate HCO3, they bind H plus less strongly than, say, OH. And here's an interesting point. Most of the acids and bases involved in the normal day-to-day regulation of acid-base balance in our extracellular fluid are actually weak ones.
That seems counterintuitive. So the weak ones are key for normal fine-tuning.
Exactly.
Okay, let's talk about measuring this H plus concentration, saying 0.000000000 equivalents per liter all the time is cumbersome.
It really is. And that's precisely why the pH scale was developed. It's a logarithmic scale designed to make these tiny numbers much more manageable.
The formula is pH as log H plus 1. Yeah.
That's the one where H plus is the concentration in equivalents per liter.
So if normal arterial blood H plus is 40 nanoequivalents per liter, which is 0.000000004 EQL,
then TH nulls is Nash log of 0.000000004, which works out to 7.4.
Got it. And crucially, it's an inverse relationship. So a low pH number means a high H plus concentration, more acidic.
Precisely. And a high pH number means a low H plus concentration, more alkaline or basic.
And these normal pH values vary a bit in different parts of the body.
They do. Arterial blood is the standard reference, typically pH 7.4. Venous blood and the interstitial fluid around cells are usually slightly lower, around 7.35.
Why is that?
It's mainly due to the extra carbon dioxide, CO2, that they pick up from tissue metabolism. CO2 forms carbonic acid in water, adding a little bit more H plus cell.
Okay. And inside the cells, intracellular fluid.
That's even more variable, generally ranging from maybe 6.0 to 7.4. It's usually lower than the fluid outside the cells because cellular metabolism itself produces acidic byproducts.
And things like low oxygen or poor blood flow can make it drop further.
Yes, absolutely. Hypoxia or ischemia tends to lower intracellular pH.
Urine pH is the most variable, you said.
Hugely variable. It can range anywhere from 4.5, quite acidic, up to 8.0, which is alkaline. This reflects the kidneys actively adjusting H plus excretion to maintain the body's overall balance.
And just for contrast, the extreme example is gastric acid.
Right, stomach acid, pure gastric HCl, can have a pH down around 0.8. That's incredibly acidic, and H plus concentration roughly 4 million times higher than blood. But of course, that's a very specialized, isolated environment.
Okay, so for the systemic circulation, normal arterial pH is 7.4. Acidemia significantly below that, alkalemia significantly above. And the limits compatible with life are roughly pH 6.8 to 8.80.
That's the generally accepted range. Outside of that, cellular function is severely compromised. It's a really narrow window for survival.
So how does the body defend this critical pH range? What are the systems?
There are 3 primary lines of defense, and they operate on different timescales, working together as a team.
First up, the chemical acid-base buffer systems.
These are the immediate responders. They're already dissolved in your body fluids and react within seconds. They combine with any excess acid or base that appears.
How do they work?
They essentially grab onto free H plus if there's too much, or release H plus if there's too little. They minimize the immediate pH change, but, and this is key, they don't actually eliminate the acid or base from the body, they just temporarily buffer it.
So they buy time. What's the second line?
That's the respiratory center. You're breathing. The system acts within minutes.
How does breathing affect pH?
Your lungs regulate the amount of CO2 you exhale. And since CO2 levels in the blood are directly linked to carbonic acid and therefore H plus concentration, simply changing your breathing rate can quickly alter your body's H plus level and pH.
Faster breathing blows off CO2, raises pH. Slower breathing lets CO2 build up, lowers pH.
You got it.
And the third line, the long-term solution.
That would be the kidneys. They are the slowest to respond, fully taking hours to even several days. But they are ultimately the most powerful regulators.
How do they provide long-term control?
The kidneys can actually eliminate excess acid or excess base, like bicarbonate, from the body by excreting acidic or alkaline urine. They directly adjust the body's total pool of bicarbonate and other substances, providing definitive long-term correction.
So it's like a relay race. Buffers take the immediate hit, lungs provide rapid adjustment, and kidneys do the long-term cleanup and rebalancing.
That's a great analogy. It has to be coordinated like that, especially considering your body produces a significant amount of acid every day just from normal metabolism, around 80 milliequivalents of what we call non-volatile acids.
80 milli-UQ a day, that has to be managed against that tiny baseline H plus concentration.
Exactly. It requires this multi-layered defense.
Okay, let's zoom in on that first line then.
Yeah.
The buffers. Can you explain the general buffer reaction again?
Sure. A buffer is basically a substance that can reversibly bind H plus DARE. You can write the reaction as buffer plus H AH buffer. If extra H plus is added, meaning things get more acidic, the reaction shifts to the right. The buffer binds the H plus plus, forming H buffer. This ties up the free H plus salt, minimizing the pH drop.
And if H plus decreases, things get more alkaline.
Then the reaction shifts to the left. H buffer releases H pluslet, replenishing the free H plus and minimizing the pH rise. The key is that the change in free H plus is much smaller than it would be without the buffer present.
This is crucial given that daily acid load and the body's sensitivity.
Absolutely critical for immediate protection.
All right, let's talk about the big one in extracellular fluid. The bicarbonate buffer system. What are its components?
It consists of two main parts: a weak acid, which is carbonic acid, H2CO3, and a bicarbonate salt, like sodium bicarbonate, NaHCO3, which provides the bicarbonate ion, HCO3, the weak base component.
How does the carbonic acid form in the first place?
It forms from the reaction between carbon dioxide and water, CO2 plus H2O, H2CO of H2CO3. This reaction happens spontaneously, but it's quite slow on its own.
But there's an enzyme.
Yes, carbonic anhydrase. This enzyme dramatically speeds up that reaction by orders of magnitude. It's found in many tissues, very importantly in lung capillary walls and in renal tubule cells.
Okay, so CO2 and water make carbonic acid quickly where needed. Then what happens to the carbonic acid?
Carbonic acid itself is a weak acid, so it only partially dissociates into H plus and bicarbonate. H2CO3 equals H plus HCO3. Meanwhile, the bicarbonate salt, like NaHCO3, dissociates almost completely in water into Na plus and HCO3.
So the whole interconnected system is really CO2 plus H2O3 equal H plus HCO3.
That's the core equilibrium.
Now, how does the system actually buffer added acid? Say a strong acid like HCl comes along.
Okay, the H plus from the strong acid, HCl, immediately reacts with the bicarbonate ion, HCO3, from the buffer system, H plus HCO3, and that's H2CO3.
So the strong, dangerous H plus gets converted into the weak acid, carbonic acid.
Exactly. You've replaced a strong acid with a much weaker one. Then, because of carbonic anhydrase, that newly formed HCO3 rapidly breaks down into CO2 and H2O.
And the extra CO2.
The increased CO2 stimulates respiration, so you breathe faster and deeper, blowing off the excess CO2. The net effect is the potentially dangerous H plus is buffered and then eliminated as CO2 via the lungs.
Very elegant. Okay, what if a strong base is added, like sodium hydroxide, NaOH?
The hydroxide ion from the NaOH reacts with the carbonic acid H2CO3 component of the buffer, OH plus H2CO3 and HCO3 plus H2O.
So the strong base is replaced by the weak base, bicarbonate.
Correct. You've neutralized the strong base. Now, as H2CO3 is consumed in this reaction, the equilibrium shifts. More CO2 and H2O will combine, again, helped by carbonic anhydrase, to replenish the H2CO3.
Does it affect breathing?
Yes. The decrease in CO2, as it's used to make H2CO3, tends to inhibit respiration slightly. Also, the reaction produces more bicarbonate, HCO3. This excess bicarbonate will eventually be dealt with by the kidneys, which can excrete it.
This dynamic relationship between H plus, HCO3, and CO2 leads us to a really important equation, doesn't it?
It does. The Henderson-Hasselbalch equation. It mathematically describes the relationship between pH, bicarbonate concentration, and the partial pressure of CO2, PSO2.
What's the equation again?
In its common form for this system, it's pH equals pK plus log, HCO3, 0.03 pco2.
Okay, let's break that down. pK.
pK is related to the dissociation constant of the weak acid, carbonic acid. For the bicarbonate system at body temperature, the pK is about 6.1.
And the HCO3 is the bicarbonate concentration.
Usually measured in milliequivalents per liter, typically around 24.
And pico2 is the partial pressure of dissolved CO2 gas.
Measured in millimeters of mercury, normally around 40 mmHg. The 0.03 factor is a solubility coefficient that converts pico2 into a molar concentration, so the units work out in the ratio.
So this equation tells us directly, if bicarbonate goes up, the log term increases, and pH goes up towards alkalosis.
Correct.
And if PCO2 goes up, the denominator increases, the log term decreases, and pH goes down towards acidosis.
Exactly. This equation is profoundly important because it shows the two key physiological variables that determine extracellular pH bicarbonate, which is mainly regulated by the kidneys, and PSO2, which is mainly regulated by the lungs.
That's the crucial link. Kidneys control bicarb, lungs control CO2. Understanding that ratio, HCO3 over PSO2, is key to diagnosing acid-base disorders like an expert.
Absolutely. It allows us to classify disorders. If the primary problem is a change in HCO3, we call it a metabolic disorder. Low HCO3 causes metabolic acidosis. High HCO3 causes metabolic alkalosis.
And if the primary problem is a change in PSO2?
That's a respiratory disorder. High PSO2 causes respiratory acidosis. Low PSO2 causes respiratory alkalosis. The Henderson-Hasselbalch equation provides the framework for understanding all of this.
Now let's revisit that pK of 6.1. A buffer is chemically most effective when the pH is close to its pK. But normal extracellular pH is 7.4, which is quite a bit higher than 6.1.
That's right. At pH 7.4, if you calculate the ratio using Henderson-Hasselbalch, you find that the concentration of HCO3 is about 20 times higher than the concentration of dissolved CO2, or H2CO3.
20 to 1. So we're operating on a part of the buffer's titration curve, where it's relatively flat. Chemically speaking, it shouldn't be the most powerful buffer per molecule at pH 7.4.
Correct. Based purely on its pK and the operating point, it doesn't seem optimally positioned for maximum chemical buffering capacity right at 7.4. Plus, its concentration isn't even that high compared to some other ions or proteins.
So why on earth is it considered the most important extracellular buffer? This seems like a paradox.
It does seem paradoxical at first glance, but here's the crucial insight, the key reason. Its power comes less from its pure chemical properties at pH 7.4 and more from the fact that its two components, HCO3 and CO2, are under constant, independent physiological control.
Controlled by the kidneys and lungs.
Precisely. The kidneys regulate the HCO3 pool, and the lungs regulate the PSO2. The body can actively adjust the levels of both components, thereby precisely controlling the ratio in the Henderson-Hasselbalch equation. It's this ability to physiologically fine-tune the buffer system that gives it such immense power and makes it the dominant player in extracellular pH regulation.
So it's not just about the chemistry, it's about the active management by other organ systems. That's a really critical distinction.
It's the absolute key takeaway for the bicarbonate system.
Okay, let's briefly touch on the phosphate buffer system. You mentioned it's important in specific places.
Right. The phosphate system involves the weak acid H2PO4 dihydrogen phosphate and its conjugate base HPO4 monohydrogen phosphate.
Same principle. Add acid, H plus binds to HPO4, forming H2PO4. Add base, OH takes H plus from H2PO4, forming HPO4.
Exactly the same buffering principle.
Now, its pK is around 6.8. That's much closer to the normal body pH of 7.4 than bicarbonate's 6.1. So it should be more effective.
Chemically, yes. Per mole, it is a more effective buffer nearer to pH 7.4 because its pK is closer. However, its overall impact in the extracellular fluid is limited by its concentration.
It's just not very concentrated out there.
Right. Phosphate concentration in the extracellular fluid is quite low, only about 8% or so of the bicarbonate concentration. So even though it's efficient per molecule, there just isn't enough of it to be the major extracellular buffer.
But you said it is important elsewhere.
Yes, very important in two key locations: the renal tubular fluid and inside cells.
Why there?
In the kidney tubules, water gets reabsorbed, which significantly concentrates the phosphate that was filtered. Also, the pH of the tubular fluid is often lower than plasma pH, sometimes getting down closer to that pK of 6.8. So higher concentration and more optimal pH make it a very effective buffer for the H plus being secreted into the urine.
And inside cells.
Phosphate concentration is considerably higher inside cells compared to outside. Plus, intracellular pH is generally lower than extracellular pH, again bringing it closer to the phosphate pK, so it plays a significant role in buffering pH changes within the cells.
Makes sense. Concentration and proximity to pK are key.
Precisely.
And the third major buffer group, proteins.
Yes, proteins are actually the most plentiful buffers in the entire body, especially within cells where they are highly concentrated.
How do they buffer?
Proteins are long chains of amino acids. Many of these amino acids have side chains that can either release or accept H plus love. Particularly important are negatively charged groups, like carboxyl groups, that can accept an H++, effectively acting as bases.
Hemoglobin was the big example you mentioned earlier.
Hemoglobin inside red blood cells is a crucial buffer. The reaction H++ HD is very important for buffering H+ generated from CO2 transport. Overall, proteins are estimated to account for something like 60 to 70% of the total chemical buffering capacity of the body fluids, mostly happening intracellularly.
Because there's just so much protein inside cells.
Exactly. High concentration equals high capacity.
But how quickly can intracellular proteins buffer changes happening in the extracellular fluid?
That's a good point. It's relatively slow, generally taking several hours for full equilibration. The reason is that H+ ions and bicarbonate ions don't cross cell membranes very easily or quickly. Red blood cells are a bit of an exception.
The CO2 crosses easily.
CO2 diffuses very rapidly across cell membranes. So changes in extracellular PCO2 will almost immediately affect intracellular pH, and intracellular buffers will respond to that quickly. But buffering extracellular H+ directly by intracellular proteins takes more time for the ions to slowly exchange or move.
So it's significant capacity, but the response time for extracellular changes isn't instantaneous, except maybe in red cells.
Correct. And again, like phosphate, many protein buffer groups have pKs that are closer to the typical intracellular pH, which enhances their effectiveness within that environment.
Okay, we've looked at bicarbonate, phosphate, and proteins. Do they operate in isolation?
Not at all. They're all intimately linked because they all exist in the same body fluids and are therefore all exposed to and in equilibrium with the same hydrogen ion concentration.
There's a principle for this.
Yes, it's called the isohydric principle.
Isohydric principle.
It basically states that because H plus is common to the equilibrium reactions of all buffer systems in a solution, any change in H plus concentration affects the balance of all the buffer systems simultaneously.
So if the bicarbonate system buffers some acid, the resulting small change in H plus instantly shifts the equilibrium of the phosphate system and the protein buffers too.
Exactly. They don't act independently. They function as an integrated network. They essentially buffer one another, shifting H plus ions back and forth between the different systems to collectively minimize the overall change in free H plus concentration.
That interconnectedness is really fundamental to the whole buffering defense. Okay, let's move to the second line of defense. The respiratory system's control via CO2.
Right. The lungs exert powerful control over acid-base balance by regulating the concentration of CO2 in the extracellular fluid.
How do they do that?
Simply by adjusting alveolar ventilation, how much air moves in and out of the alveoli where gas exchange happens. Your body constantly produces CO2 through metabolism. This CO2 is transported to the lungs and exhaled. Normally, the rate of CO2 elimination by the lungs matches its rate of production, keeping the partial pressure of CO2, the physico, stable around 40 millimol Hg.
And how does changing ventilation affect H plus speed?
It's a direct consequence of the bicarbonate buffer equilibrium. CO2 plus H2O equals H2CO3 equals H plus HCO3. If you increase your ventilation rate, you blow off more CO2. This lowers the PiCO2 in your blood.
Less CO2 shifts the equilibrium to the left.
Exactly. It pulls the equilibrium to the left, consuming H plus and H2CO3. So, look, PiCO2 leads to lower air plus concentration, and the pH rises, becomes more alkaline.
And if you decrease ventilation?
If you hypoventilate, CO2 isn't eliminated as effectively. It builds up in the blood. PiCO2 rises. This pushes the equilibrium to the right, generating more H2CO3 and releasing more H plus fundus. So higher PiCO2 leads to higher H plus concentration, and the pH falls, becomes more acidic.
So your breathing rate acts like a rapid control knob for blood pH.
A very powerful and rapid one. The source material mentions that doubling alveolar ventilation can raise blood pH by about 0.23 units fairly quickly. Conversely, decreasing ventilation to just 1/4 of normal can lower pH by about 0.45 units, so the lungs can induce significant pH changes.
Is this under automatic feedback control? Does the body sense pH and adjust breathing accordingly?
Oh, absolutely. This is a crucial negative feedback loop. The respiratory center in the brainstem is sensitive to changes in H plus concentration and also directly to PiCO2 in the blood and cerebrospinal fluid.
So if H plus increases, pH drops.
That stimulates the respiratory center, causing you to breathe more deeply and more rapidly hyperventilation. This increased ventilation blows off more CO2, lowers PiCO2, and helps bring the H plus concentration back down towards normal, thus raising the pH.
And the opposite, if H plus falls, pH rises.
If H plus falls, alkalosis, the respiratory center is depressed. Ventilation decreases, hypoventilation. This allows CO2 to accumulate, raising PiCO2, which pushes the equilibrium rate, increasing H plus concentration back towards normal and lowering the pH.
Is the response equal in both directions?
Not quite. The response to decreased ventilation in alkalosis is somewhat limited. If you decrease breathing too much, oxygen levels in the blood start to fall, hypoxemia. Low oxygen is a powerful stimulus for breathing via different receptors, peripheral chemoreceptors. So the need for oxygen can override the signal to slow down breathing due to alkalosis. Therefore, the respiratory compensation is generally more robust for acidosis than for alkalosis.
How effective is this respiratory control system at correcting pH disturbances that don't originate in the lungs themselves, like metabolic acidosis?
It's quite effective as a rapid compensatory mechanism. Estimates suggest it can correct about 50 to 75% of the initial pH disturbance, meaning it has a feedback gain of about 1 to 3, and it acts fast. Significant adjustments happen within 3 to 12 minutes.
So it's a powerful physiological buffer.
Exactly. It's not a chemical buffer like bicarbonate, but a physiological one. Its total buffering power is estimated to be 1 to 2 times greater than all the chemical buffers in the extracellular fluid combined. It rapidly stabilizes pH, buying crucial time until the slower but more definitive kidneys can act.
But of course, if the lungs themselves are the problem.
Right. If lung function is impaired, like in severe emphysema or pneumonia, then the ability of the respiratory system to compensate for a metabolic acid-base problem is severely compromised. Impaired lungs might even cause a respiratory acidosis by retaining CO2. In such cases, the kidneys become even more critical as the main remaining line of physiological defense.
Which brings us neatly to the third line of defense,
Oh.
the kidneys, the long-term powerhouses.
Indeed, the kidneys provide the ultimate long-term control over acid-base balance. They do this by controlling the excretion of acids and bases in the urine.
How do they control excretion?
Fundamentally, they can excrete either an acidic urine or an alkaline urine. Excreting acidic urine removes net acid from the blood. Excreting alkaline urine removes net base, primarily bicarbonate, from the blood. By adjusting the composition of the urine, they fine-tune the body's overall acid-base status.
And the sheer volume of buffer they handle daily is staggering, isn't it? The bicarbonate filtering.
It's enormous. Every day, the glomeruli in your kidneys filter roughly 400 and 320 milliequivalents of bicarbonate out of the blood plasma and into the tubular fluid.
4320 MEQ.
Yes. Now compare that to the roughly 80 millieq of non-volatile acids produced daily by metabolism. You can see that simply preventing the loss of that filtered bicarbonate is quantitatively a much bigger job for the kidneys than excreting the daily metabolic acid load.
So job number 1 is reabsorbing almost all of that filtered bicarbonate.
Under normal conditions, yes. They reabsorb virtually all 4,320 NOHs. And the fascinating thing is that both this massive bicarbonate reabsorption and the excretion of the 80 mmHg of acid are achieved through the same basic mechanism, secretion of hydrogen ions, H+, by the cells lining the renal tubules.
So H+ secretion is the key process for both tasks.
It's the central mechanism. To reabsorb the 4,320 milliQ of filtered HCO3, the tubules must secrete at least 4,320 milliQ of H+ into the tubular fluid to react with it. Then, to get rid of the 80 milliQ of non-volatile acid, they need to secrete an additional 80 milliQ of H+.
So total daily H+ secretion under normal conditions is around 4,400 milliQ. That's huge.
It is, and the kidneys precisely regulate this massive secretion rate based on the body's needs.
How do they adjust in alkalosis when the body has too much base or too little acid?
In alkalosis, the kidneys decrease their rate of H+ secretion. This means they don't secrete enough H+ to react with all the bicarbonate that was filtered. As a result, they fail to reabsorb all the filtered HCO3, and the excess bicarbonate is simply left behind in the tubular fluid and excreted in the urine.
Losing bicarbonate in the urine is like adding acid to the blood, right?
Yeah, exactly. It helps correct the alkalosis by removing base.
And in acidosis, when there's too much acid?
In acidosis, the kidneys dramatically increase H+ secretion. They secrete far more H+ than is needed just to reabsorb the filtered bicarbonate. This ensures that all filtered bicarbonate is reabsorbed, preventing any loss. And critically, the extra secreted H+ is then used to generate new bicarbonate, which is added back to the blood.
New bicarbonate. Explain that again.
This is a crucial concept. When H+ is secreted into the tubule, it originates from the splitting of H2CO3 inside the tubular cell, which also produces an HCO3. If the secreted H+ combines with filtered HCO3 in the lumen, the intracellular HCO3 simply replaces the one that was filtered, no net gain. But if the secreted H+ combines with other buffers in the urine, like phosphate or ammonia, the intracellular HCO3 that gets reabsorbed into the blood represents a net gain of bicarbonate for the body. This new bicarbonate helps replenish the buffer stores depleted by the acidosis.
Okay, so the three fundamental renal jobs are H+ secretion, reabsorbing filtered HCO3, and producing new HCO3, and they're all tied to H+ secretion.
That's the framework.
Let's break down where and how this H+ secretion and HCO3 reabsorption happens along the tubule.
Okay. H+ secretion and HCO3 reabsorption occur in almost all segments of the tubule, except for the thin descending and thin ascending limbs of the loop of Henle. The vast majority, about 80–90% of the filtered bicarbonate, is reabsorbed in the proximal tubule.
80–90% right at the beginning.
Yes, it's a massive amount. Another 10% or so is reabsorbed in the thick ascending limb of the loop of Henle, and the remaining small amount is handled by the distal tubules and collecting ducts, where fine-tuning occurs.
How is H+ secreted in those early segments? The proximal tubule, thick ascending limb, early distal tubule. What's the mechanism?
In these earlier parts, H+ secretion is primarily driven by secondary active transport. The main player is a protein on the luminal membrane facing the tubular fluid called the sodium-hydrogen exchanger, or NHE.
Sodium-hydrogen exchanger. How does it work?
It uses the energy stored in the sodium concentration gradient. Sodium concentration is high in the tubular fluid and low inside the cell. So sodium naturally wants to move into the cell. The NHE allows sodium to move downhill into the cell and uses the energy released from that movement to pump H plus uphill out of the cell into the tubular lumen.
So it swaps one A plus coming in for one H plus going out.
Mm.
Where does the energy ultimately come from?
The energy comes from the sodium-potassium ATPase pump, located on the other side of the cell, the basolateral membrane facing the blood. This pump constantly uses ATP to pump sodium out of the cell, keeping the intracellular sodium concentration low, and thus maintaining the gradient that drives the NHE on the luminal side. It's indirect energy use for H plus secretion.
Okay, that makes sense. Now, how does this H plus secretion lead to bicarbonate reabsorption? Where do the H plus and HCO3 inside the cell come from?
Inside the tubular cell, carbon dioxide, either from cell metabolism or diffused in from the lumen blood, combines with water. This reaction, CO2 plus H2O, HH2CO3, is catalyzed by carbonic anhydrase within the cell. The H2CO3 then spontaneously dissociates into H plus and HCO3.
And the H plus is the one that gets secreted by the NHE.
Exactly. The H plus generated inside the cell is pumped out into the lumen by the NHE. The HCO3 that was generated alongside it inside the cell is then transported across the basolateral membrane into the interstitial fluid and blood. This basolateral transport often involves cotransport with sodium or exchange with chloride.
So H plus goes out to lumen, HCO3 goes towards the blood. But you said this reabsorbs the filtered HCO3. How does that connect? The HCO3 going into the blood was made inside the cell.
This is the clever part. The HCO3 that was originally filtered into the lumen cannot be directly transported across the luminal membrane into the cell. Instead, the H plus that gets secreted into the lumen immediately combines with that filtered HCO3 present in the tubular fluid.
H plus HCO3, H2CO3 in the lumen.
Right. And then often with the help of carbonic anhydrase located on the brush border of the proximal tubule cells, that H2CO3 in the lumen quickly dehydrates into CO2 and H2O.
And CO2 can cross the membrane easily.
Very easily. CO2 is lipid soluble and diffuses readily across the luminal membrane into the tubular cell. Once inside the cell, that same CO2 molecule combines with water, using intracellular carbonic anhydrase, to form H2CO3 again, which then splits into H plus and a new HCO3.
So the H plus gets secreted again, and this new intracellular HCO3 is the one that gets transported into the blood.
Precisely. So although the specific bicarbonate molecule that ends up in the blood wasn't the one originally filtered, the net effect is the same. For every H plus secreted that combines with filtered HCO3, one HCO3 disappears from the lumen and one HCO3 appears in the blood. It's an effective reabsorption process, even though it's indirect.
That's a really neat mechanism. It effectively swaps the liminal HCO3 for an intracellularly generated one that can then move to the blood.
Exactly. And notice, because the secreted H plus is immediately consumed by reacting with filtered HCO3, there's no significant net secretion or excretion of H plus from the body via this mechanism in the early tubules. Its primary role here is just reclaiming that vital filtered bicarbonate buffer.
Does this secondary active transport system have limits on how much it can acidify the urine?
Yes. The sodium-hydrogen exchanger can only establish a certain concentration gradient for H plus ties. It can typically lower the tubular fluid pH down to about 6.7, but not much further. It's great for moving large volumes against a small gradient, reabsorbing the bulk bicarbonate, but not for creating highly acidic urine.
For that, we need the later segments, late distal tubule and collecting ducts.
Correct. Starting in the late distal tubule and continuing through the collecting ducts, a different mechanism takes over for more powerful H plus secretion. This involves primary active transport carried out by specialized cells called type A intercalated cells.
Type A intercalated cells. Primary active transport means they use ATP directly.
Yes. These cells have proton pumps specifically, an H plus ATPase and often an H plus K plus ATPase located on their luminal membrane. These pumps use the energy released from splitting ATP to directly pump H plus from the cell into the tubular lumen, even against a very steep concentration gradient.
Is the process inside the cell similar, making H plus and HCO3 from CO2 and water?
Yes, the intracellular generation is the same. CO2 plus H2O minus 2CO3 minus H plus HCO3 catalyzed by carbonic anhydrase. The H plus produced is then actively pumped into the lumen by the ATP-driven pumps. The HCO3 produced is transported out across the basolateral membrane into the blood, usually in exchange for chloride. So just like before, For every H plus secreted, 1 HCO3 is reabsorbed into the blood.
But the key difference is the power of the pump.
Exactly. Because these pumps use ATP directly, they can generate a much larger H plus concentration gradient, up to about 900-fold difference between the cell and the lumen. This allows them to lower the tubular fluid pH all the way down to the minimum possible urine pH, around 4.5.
So even though this primary active transport accounts for only maybe 5% of the total H plus secreted by the kidney,
It's absolutely essential for excreting the daily acid load, especially during acidosis, because it's the only mechanism that can create maximally acidic urine.
okay, so H plus secretion reclaims filtered bicarbonate using both secondary and primary transport, and the primary active transport can also create very acidic urine. But you said earlier we can't excrete the daily 80 millieq acid load just as free H plus pri. The urine volume would be impossible.
Correct. Even at the minimum pH of 4.5, the free H plus concentration is only about 0.03 millieq per liter. To excrete 80 millieq that way would require over 2,600 liters of urine per day. Clearly, that's not how it works.
So the vast majority of that 80 millieq acid load must be excreted by combining the secreted H plus with buffers present in the tubular fluid.
Exactly. The secreted H plus, beyond what's needed to reabsorb bicarbonate, combines with urinary buffers. The two main ones are the phosphate buffer system and the ammonia buffer system.
And this process of buffering H plus in the lumen is linked to generating that new bicarbonate we talked about.
Yes, this is the crucial connection. Remember, every time an H plus is secreted, originating from H2CO3 inside the cell, an HCO3 is also generated inside the cell. When that secreted H plus combines with a buffer in the lumen other than filtered bicarbonate like phosphate, HPO4 or ammonia, NH3, the HCO3 generated inside the cell represents a net gain of bicarbonate for the body. It gets transported into the blood and helps replenish the body's buffer stores, which is essential for correcting acidosis.
Got it. So H plus secreted plus urinary buffered H plus excreted in urine. Meanwhile, the corresponding intracellularly generated HPO3 added to blood as new bicarbonate.
That's the mechanism. Let's look at the phosphate buffer first.
Okay, phosphate HPO4 in the tubules.
As we discussed, phosphate gets concentrated in the tubules as water is reabsorbed. Its pK of 6.8 is also quite effective in the typical pH range of tubular fluid, which is often lower than 7.4.
So how does it buffer the secreted H plus?
The secreted H plus enters the lumen and combines with the base form of the phosphate buffer, HPO4, to form the acid form, H2PO4. This H2PO4 molecule then travels down the tubule and is excreted in the urine, carrying the excess H plus with it, usually as a salt like sodium dihydrogen phosphate, NaH2PO4.
And for every H plus excreted this way bound to phosphate,
A new bicarbonate molecule generated inside the tubular cell during the H plus secretion process is transported into the blood.
okay, so phosphate buffering contributes to acid excretion and new bicarb generation. But is there enough phosphate to handle the whole load?
Not usually. The amount of phosphate filtered each day is relatively fixed and limits the amount of H plus that can be buffered this way, typically only about 30 to 40 millieq per day. While important, this isn't sufficient to handle large acid loads or significant acidosis.
So the other major buffer system must be more adaptable. That's the ammonia system.
Yes, the ammonia buffer system involving ammonia, NH3, and the ammonium ion, NH4+, is quantitatively much more important, especially its ability to ramp up during acidosis.
Where does the ammonia come from?
The primary source is the metabolism of the amino acid glutamine within the renal tubular cells themselves. This happens mainly in the proximal tubules, but also in the thick ascending limb and distal tubules.
How does glutamine metabolism produce ammonia and contribute to acid excretion?
It's a bit complex, but the key steps are shown in the source material, figures 31-8, 31-9. In the proximal tubule cells, one molecule of glutamine is metabolized to produce two ammonium ions, NH4+, and two bicarbonate ions, HCO3.
Two NH4 and two HCO3 from one glutamine.
Correct. The NH4+ is then actively transported into the tubular lumen, often using that same sodium-hydrogen exchanger, NHE, but operating in an NH4 plus Na plus exchange mode. The two HCO3 molecules generated inside the cell are transported across the basolateral membrane into the blood.
So right there in the proximal tubule, glutamine metabolism directly leads to NH4 plus secretion into the urine and the addition of new HCO3 to the blood. Two new bicarbs for every glutamine used.
Exactly, that's a major pathway. But the collecting ducts also play a critical role, particularly in concentrating the final urine.
How does it work there?
In the collecting duct cells, ammonia, NH3, which is also produced from glutamine metabolism further up or diffuses from the interstitium, can readily diffuse across the cell membranes into the tubular lumen because NH3 is lipid-soluble.
So NH3 itself moves into the lumen. Then what?
In the lumen, this NH3 encounters the H+ that is being actively secreted by the type A intercalated cells using those primary active transport pumps, H+ NH-ATPase. The H+ combines with the NH3 to form the ammonium ion, H+ plus NH3, NH3, and NH4 plus, right?
And NH4 plus is an ion, so it's not lipid soluble.
Right. NH4 plus is much less permeable across the membrane than NH3. So once formed in the lumen, it's effectively trapped there and gets excreted in the urine.
And the benefit of trapping it.
Every time an H+ secreted by the collecting duct cell combines with NH3 to form trapped NH4 plus ray, the corresponding HCO3 that was generated inside the cell during H+ secretion gets transported into the blood as new bicarbonate.
So both proximal glutamine breakdown generating NH4 plus directly and collecting duct trapping of NH3 as NH4 plus 3 contribute to ammonia excretion and new bicarbonate generation.
They do. And the ammonia system has a crucial regulatory advantage over the phosphate system. Its activity is highly responsive to the body's acid-base status.
How so?
Increased extracellular H+ concentration acidosis strongly stimulates the kidneys to increase their rate of glutamine metabolism and subsequent NH4 plus production and excretion.
So in acidosis, the kidneys make and excrete much more ammonium.
Dramatically more. Under normal conditions, the ammonia system might account for roughly half of the daily acid excretion and new bicarbonate generation, maybe 30-40 milliliters a day. But in chronic acidosis, the kidneys can ramp up NH4 plus excretion enormously, sometimes tenfold or more, potentially reaching up to 500 milliliters per day.
500 ml a day?
Yes. This makes the ammonia buffer system the overwhelmingly dominant mechanism for eliminating large acid loads from the body and generating the large amounts of new bicarbonate needed to restore balance during chronic acidosis. That adaptive capacity is absolutely key.
Wow. Okay, so how do we quantify the overall net effect of all these kidney processes, bicarbonate reabsorption, H+ buffered by phosphate, H+ buffered by ammonia, and any bicarbonate that might be excreted?
We use a measurement called net acid excretion, or NAE. It sums up the kidney's total contribution to acid removal from the body.
What's the formula for NAE?
Net acid excretion equals NH4 plus excretion plus urinary titratable acid, HCO3 excretion.
Well, let's break that down. NH4 plus excretion is just the amount of ammonia measured in the urine.
Correct. That represents the H+ excreted bound to ammonia.
Urinary titratable acid, what's that?
Titratable acid is measured by taking a urine sample and adding a strong base, like NaOH, until the urine pH returns to 7.4, the normal pH of plasma. The amount of base needed equals the amount of H+ that was present in the urine bound to buffers, other than ammonia, primarily phosphate, H2PO4, but also minor buffers like urate and citrate. It's essentially H+ excreted with non-bicarbonate, non-ammonia buffers.
Okay, and why do we subtract HCO3 excretion?
Because losing bicarbonate in the urine has the same physiological effect as gaining H+ in the blood, it removes base from the body, making the body more acidic. So HCO3 excretion counts against net acid excretion.
So to maintain perfect acid-base balance day to day, the net acid excretion must exactly equal the amount of non-volatile acid produced by metabolism.
Precisely. Normally, NAE needs to be around 80 ml a day to match the typical metabolic acid production.
How does NAE change in acidosis or alkalosis?
In acidosis, the kidneys work overtime to get rid of acid. NAE increases significantly, often rising well above 80 ml a day. This increase is driven primarily by a huge surge in NH4 plus excretion and also some increase in titratable acid. Bicarbonate excretion drops to zero. The result is removal of excess acid and a net addition of new bicarbonate to the blood.
When in alkalosis?
The situation reverses. The kidneys need to retain acid and get rid of base. H+ secretion plummets. Since there's no excess H+ being secreted beyond what's needed for bicarb reabsorption, and even that might be incomplete, titratable acid and NH4 plus excretion drop essentially to zero. Meanwhile, bicarbonate excretion increases dramatically as the kidneys fail to reabsorb all the filtered load. This results in a negative net acid excretion, meaning the kidneys are causing a net loss of bicarbonate base from the body.
And all of this adaptability hinges on the precise regulation of that initial H+ secretion step by the tubular cells.
Exactly. The rate of H+ secretion is the master control point. Normally, it's finely tuned to ensure just enough H plus is secreted to reabsorb all the filtered bicarbonate plus excrete the daily 80 milliQ acid load as titratable acid and ammonia.
So to recap, in alkalosis, H plus secretion rate drops.
Yes, this leads to incomplete HCO3 reabsorption, so HCO3 is lost in urine, and virtually no H plus left over to form titratable acid or NH4 plus net effect, base loss, no new base generated.
And in acidosis, H plus secretion rate ramps up.
Dramatically. This ensures complete HCO3 reabsorption, conserving base, and provides a large amount of excess H plus to be buffered by phosphate and especially ammonia, leading to high rates of titratable acid and NH4 plus excretion. Net effect: acid removal, large generation of new base HCO3.
What are the main physiological signals that tell the kidney tubule cells to increase H plus secretion during acidosis?
The two most important direct stimuli are an increase in the pico-2 of the extracellular fluid, which occurs in respiratory acidosis, and an increase in the H plus concentration itself, a decrease in pH, which occurs in both respiratory and metabolic acidosis. These directly stimulate the H plus secretory mechanisms in the tubular cells.
And conversely, what signals decrease H plus secretion in alkalosis?
A decrease in extracellular pico-2 in respiratory alkalosis, and a decrease in extracellular H plus concentration, increase in pH, seen in both respiratory and metabolic alkalosis. These directly inhibit H plus secretion.
The source material also mentions other factors influencing H plus secretion and bicarbonate reabsorption, like hormones and electrolytes. Table 31-2 concept.
Yes, several other factors modulate the process. Aldosterone is a key one.
Aldosterone, the salt-retaining hormone.
Yes, but it also directly stimulates H plus secretion by the type A intercalated cells in the collecting ducts. So conditions with excess aldosterone, like Conn syndrome, can lead to increased H plus loss, increased bicarbonate reabsorption, and potentially cause metabolic alkalosis.
What about body fluid volume?
Extracellular fluid volume depletion is another important factor. When volume is low, the body tries hard to retain sodium. Many sodium reabsorption mechanisms in the tubules are linked, directly or indirectly, to H plus secretion, like the Na plus H plus exchanger. Also, volume depletion stimulates the renin-angiotensin-aldosterone system. Angiotensin II itself stimulates the Na plus H plus exchanger, and aldosterone stimulates H plus secretion. So volume depletion often leads to increased H plus secretion and bicarbonate reabsorption, creating a tendency towards metabolic alkalosis.
Interesting connection. And potassium levels.
Plasma potassium concentration has a significant impact. Hypokalemia, low potassium, tends to stimulate H plus secretion and bicarbonate reabsorption, potentially leading to or worsening metabolic alkalosis. This is partly because low extracellular K plus causes K plus to move out of cells, and H plus moves into cells to maintain electroneutrality, making the tubular cells more acidic and stimulating H plus secretion.
And high potassium. Hyperkalemia.
Hyperkalemia has the opposite effect. It tends to inhibit H plus secretion and bicarbonate reabsorption, contributing to metabolic acidosis.
Okay, we've dissected the components: buffers, lungs, kidneys, and their regulation. Let's try to integrate this now and look at how the kidneys specifically contribute to correcting the major acid-base disorders.
Right. Let's consider acidosis first. pH is below 7.4, meaning the HCO3 PSO2 ratio is too low.
Scenario 1. Metabolic acidosis. The primary problem is low bicarbonate HCO3.
Here, the kidneys are the main long-term defense. They go into overdrive to correct this. They will reabsorb all the filtered bicarbonate to prevent any further loss. And crucially, they dramatically increase their production and addition of new bicarbonate to the blood by maximizing titratable acid excretion and especially ramping up ammonium NH4 plus excretion.
And the lungs help out quickly.
Yes, the low pH stimulates the respiratory center, causing hyperventilation. This lowers PCO2, which helps bring the HCO3 PCO2 ratio back up towards normal. That's the respiratory compensation.
Scenario 2. Respiratory acidosis. Primary problem is high PCO2, usually due to inadequate ventilation.
The initial hit is taken by the chemical buffers, but the real physiological correction comes from the kidneys over the next few days. The high PCO2 and resulting low pH stimulate the tubular cells to increase H plus secretion. This leads to complete reabsorption of filtered bicarbonate and significant generation of new bicarbonate. Plasma HCO3 levels rise.
Raising HCO3 helps normalize the HCO3 PCO2 ratio, even though PCO2 is still high.
Exactly. It compensates for the respiratory problem by creating a partially offsetting metabolic alkalosis. This renal compensation takes time, though typically 3 to 5 days to reach its maximum effect, as shown in Table 31-3.
Okay, now let's look at alkalosis, where pH is above 7.4 and the HCO3 PCO2 ratio is too high.
Scenario 3. Metabolic alkalosis. Primary problem is high bicarbonate HCO3. Here, the kidneys need to get rid of the excess space. They do this by decreasing H+ secretion. Since less H+ is secreted, it's not enough to react with all the filtered bicarbonate, which is already high. The excess, unreabsorbed HCO3 is excreted in the urine, removing base from the body.
And the lungs compensate.
Yes, the high pH tends to suppress respiration slightly, causing hypoventilation. This raises PCO2, which helps lower the high HCO3 PCO2 ratio back towards normal. Respiratory compensation for metabolic alkalosis.
Scenario 4, respiratory alkalosis. Primary problem is low PCO2, usually from hyperventilation.
Again, chemical buffers provide some immediate damping. The kidneys then compensate over days. The low PCO2 and high pH inhibit tubular H+ secretion. Less H+ secretion means less bicarbonate is reabsorbed, more HCO3 is excreted in the urine, lowering the plasma HCO3 concentration.
Lowering HCO3 helps bring the high HCO3 PCO2 ratio back down towards normal.
Precisely. It's renal compensation for respiratory alkalosis, again taking a few days to fully develop. The summary table in the source, Table 31-3, is really useful for seeing these patterns of primary change and expected compensation for pH, PCO2, and HCO3 in all 4 simple disorders.
Absolutely. It's a great quick reference for what you'd expect to see. Okay, let's shift gears slightly towards the clinical side, thinking about causes, diagnosis, and treatment. What typically causes respiratory acidosis?
Anything that impairs the lungs' ability to get rid of CO2, leading to a buildup of PCO2. Common causes include damage to the respiratory control center in the brain, severe airway obstruction, like in an asthma attack or advanced COPD, pneumonia that affects large areas of the lung, conditions like emphysema that destroy gas exchange surface area, or anything else that reduces effective ventilation.
Respiratory alkalosis is the opposite, blowing off too much CO2.
Right. Excessive ventilation leading to low PCO2. Often, the cause isn't primarily a lung disease. Psychological hyperventilation due to anxiety or panic is very common. Another classic cause is the physiological response to high altitude, where low oxygen stimulates breathing, driving off CO2.
Metabolic acidosis.
Yeah.
The core problem is low extracellular bicarbonate. The sources group the causes into a few general categories.
Yes, broadly speaking, you get metabolic acidosis. 1, the kidneys fail to excrete the metabolic acids produced daily. 2, the body produces an excessive amount of metabolic acids. 3, you ingest acids or substances metabolized to acids. Or 4, you lose excessive amounts of base, bicarbonate, from the body.
Can you give some specific examples?
Sure. Kidney failure itself, chronic renal failure, is a major cause due to impaired acid excretion and reduced new bicarb generation. Specific kidney defects like renal tubular acidosis, RTA, impair H+ secretion or HCO3 reabsorption. Severe diarrhea is perhaps the most common cause worldwide due to large losses of bicarbonate-rich fluid from the intestines.
What else?
Vomiting of intestinal contents, which are alkaline, can also cause bicarbonate loss. Diabetic ketoacidosis involves massive overproduction of keto acids. Ingesting poisons like aspirin, salicylic acid, methanol, metabolized to formic acid, or ethylene glycol, metabolized to glycolic and oxalic acids, causes severe acidosis.
And metabolic alkalosis. The problem is high extracellular bicarbonate.
This usually results from either excessive retention of bicarbonate or, more commonly, excessive loss of hydrogen ions from the body. Classic causes include the use of certain diuretics, like loop diuretics or thiazides, which increase renal H+ secretion indirectly by affecting sodium and potassium transport.
Aldosterone again.
Yes, conditions with excess aldosterone directly stimulate renal H+ secretion, leading to H+ loss and bicarbonate retention. Another very common cause is vomiting of gastric contents. Only losing large amounts of hydrochloric acid, HCl, from the stomach leads directly to alkalosis. Also, ingesting large amounts of alkaline substances like sodium bicarbonate, baking soda, can cause it.
If a patient presents with one of these significant acid-based disturbances, how is it typically treated?
Well, the absolute best treatment is always to identify and correct the underlying cause, if possible. Treating the pneumonia causing respiratory acidosis, managing the diabetes causing ketoacidosis, stopping the drug causing the problem, etc.
But sometimes you need to directly address the pH imbalance itself.
Yes, especially if it's severe or the underlying cause can't be fixed quickly. For severe metabolic acidosis, you might need to administer bicarbonate to neutralize the excess acid. This can be given orally as sodium bicarbonate or, more acutely, intravenously.
IV bicarbonate needs caution.
Yes, it has potential risks like volume overload, hypernatremia, and potentially worsening intracellular acidosis in some situations, so it must be used carefully. Sometimes substances like sodium lactate or sodium gluconate are used IV. The body metabolizes the lactate or gluconate, consuming H+, and effectively generating bicarbonate.
And for neutralizing excess base in metabolic alkalosis.
Agents like ammonium chloride can be given orally. The ammonium, NH4+, is metabolized by the liver to urea, which releases HCl, helping to neutralize the excess base. 4V ammonium chloride is rarely used due to toxicity risks. Often, for metabolic alkalosis associated with volume depletion, simply giving IV saline solution is effective treatment, as correcting the volume deficit removes the stimuli for renal H+ retention.
How do clinicians actually diagnose these disorders when a patient comes in? What measurements are key?
The cornerstone of diagnosis is analyzing an arterial blood gas, ABG, sample. This gives you 3 critical values: the blood pH, the plasma bicarbonate concentration, HCO3, and the partial pressure of carbon dioxide, and PCO2.
And there's a systematic way to interpret these numbers.
Yes, the sources outline a clear step-by-step approach, similar to Figure 3110. First, look at the pH. Is it less than 7.4 acidosis or greater than 7.4 alkalosis?
Step 1. Acidosis or alkalosis.
Step 2. Look at the PO2. Normal is about 40 millimole Hg, and the HCO3 normal is about 24 MEQL. Which one is abnormal in a direction that would explain the pH change?
Step 3. Identify the primary disturbance.
Exactly. If the PCO2 change explains the pH, e.g. low pH with high PCO2, the primary problem is likely respiratory. If the HCO3 change explains the pH, e.g. low pH with low HCO3, the primary problem is likely metabolic.
Step 4. Check for compensation.
Right. Once you've identified the likely primary disturbance, look at the other value. Is it changing in the direction you'd expect for compensation? For example, in primary metabolic acidosis, low pH, low HCO3, you expect respiratory compensation, so PCO2 should also be low. In primary respiratory acidosis, low pH, high PCO2, you expect renal compensation, so HCO3 should be high, or at least higher than baseline if it's acute.
What if the compensation doesn't seem appropriate, or both PCO2 and HCO3 are abnormal in ways that don't fit a simple pattern?
That suggests a complex or mixed acid-base disorder where there might be 2 or more primary disturbances occurring simultaneously. For instance, a patient could have both metabolic acidosis, low HCO3, and respiratory acidosis, high PCO2, at the same time, leading to a very low pH. Interpreting these mixed disorders requires careful consideration of the clinical context and the magnitude of the changes.
Is the acid-base nomogram commonly used for this?
Yes, nomograms like the one shown, Figure 3111 concept, are very helpful visual tools. They plot pH, PCO2, and HCO3 simultaneously. There's a central area representing the normal range. Then there are shaded bands radiating outwards that show the typical range of values expected for each of the 4 simple acid-base disorders with appropriate compensation, assuming enough time has passed for that compensation to occur. A few hours for respiratory compensation, a few days for full renal compensation.
How do you use it?
You plot the patient's pH, PCO2, and HCO3 values on the nomogram. If the point falls clearly within one of the shaded bands, it strongly suggests that simple disorder with appropriate compensation. If the point falls outside any of the expected compensatory bands, it's a strong indicator of a mixed disorder or potentially inadequate compensation for some reason. It's a quick check, but always needs to be interpreted alongside the patient's clinical picture.
Okay, one last diagnostic tool mentioned. The anion gap. How does that help?
The anion gap is a really useful concept, particularly for figuring out the underlying cause of a metabolic acidosis.
How is it calculated?
It's calculated from the commonly measured electrolytes in the plasma. Anion gap, sodium concentration, chloride concentration plus bicarbonate concentration.
Sodium minus chloride plus bicarb.
Right. Normally, the measured cations, mainly sodium, exceed the measured anions, chloride and bicarb. This gap represents the concentration of unmeasured anions, things like plasma proteins, which carry a net negative charge, phosphate, sulfate, and organic anions. The normal anion gap is typically in the range of 8 to 16 MEQL.
So how does this help specifically with metabolic acidosis, where bicarbonate is low?
When bicarbonate goes down in metabolic acidosis, something else has to change chemically to maintain electrical neutrality in the plasma. Either chloride levels must go up to replace the lost negative charge of bicarbonate, or the levels of those unmeasured anions must go up. The anion gap helps distinguish between these 2 scenarios.
Scenario 1. normal anion gap metabolic acidosis.
This is also called hyperchloremic metabolic acidosis. Here, the fall in bicarbonate is matched by a roughly equal increase in chloride concentration. Since chloride is measured in the anion gap calculation, the gap itself remains normal. This pattern typically occurs when bicarbonate is lost directly from the body, like in diarrhea, or in certain types of renal tubular acidosis where the kidney inappropriately loses bicarbonate or retains chloride.
Scenario 2. Increased anion gap metabolic acidosis.
This occurs when the fall in bicarbonate is not matched by an increase in chloride. Instead, the reason bicarbonate is low is because it has been consumed buffering an accumulation of some other acid whose anion is not typically measured, that is, it's one of the unmeasured anions. These accumulated unmeasured anions widen the gap between measured chemations and measured anions.
So a high anion gap points towards accumulation of specific types of acids.
Exactly. It strongly suggests the presence of excess unmeasured acid anions. Classic causes of high anion gap metabolic acidosis include diabetic ketoacidosis, accumulation of ketoacetate and beta-hydroxybutyrate anions, lactic acidosis, accumulation of lactate, advanced chronic renal failure, accumulation of phosphate, sulfate, and other organic anions that the failing kidneys can't excrete, and certain poisonings like aspirin, methanol, or ethylene glycol, where toxic acid metabolites accumulate. Starvation can also cause a mild ketoacidosis with an increased gap. It's a very powerful diagnostic clue for narrowing down the cause of metabolic acidosis. Table 1 to 4 gives examples.
This whole picture, the instantaneous chemical buffers, the rapid adjustments by the lungs controlling CO2, and the incredibly sophisticated work of the kidneys reclaiming bicarbonate, secreting H+O, dealing with phosphate and ammonia, and generating new bicarbonate, it's just an amazing symphony of physiological regulation.
It truly is remarkable, the level of coordination and adaptability required to maintain hydrogen ion concentration within that incredibly narrow range, despite constant challenges from metabolism and the environment, is just a testament to the elegance of biological systems.
And mastering the details, as we've tried to do today, really gives you a profound appreciation for that core physiology and, of course, why things go wrong in disease.
Absolutely. You really see how interconnected the renal, respiratory, and metabolic systems truly are. You can't understand acid-base balance by looking at them in isolation.
Definitely not. So we've seen how the body pulls out all the stops, from a microscopic pumps in kidney cells to adjusting your very breathing just to keep that delicate pH balance. It leaves you thinking, doesn't it? What happens when multiple systems are failing simultaneously, like in a patient with both severe kidney disease and severe lung disease? How does the body even attempt to prioritize? How do clinicians intervene? And ultimately, what are the absolute limits of this extraordinary physiological control? That interplay in complex illness is certainly something to mull over.